Thank you very much. It's great to be in Hyderabad. I can say that the weather here is better than the weather in my hometown of Houston, Texas right now. And so that is a welcome surprise. And I also want to, before starting my presentation, say thank you very much to the kind students who picked me up at 4 a.m. at the airport. So I remember being a graduate student, and thanks to their efforts, I made it here safely, and appreciate them staying up late to come do that. So if we move forward, so I'm going to speak today about development of our lead product, which is a personalized tumor vaccine that incorporates our proprietary biological adjuvant proteins. And I'll tell you what those are in a bit. I want to say that perhaps to tie the prior presentation and my presentation together in a theme, the key point is you can develop any technology in the laboratory, but if you want to take it to the market, and particularly to the patients, to help the patients in dire need with terminal cancer, you have to be able to manufacture it. So that is one of the things that we have focused on extensively with our product, is to make the logistics and manufacturing of a personalized therapy for cancer more doable, simpler, lowering the cost of goods and expanding the market opportunity for all patients, including hopefully patients in India. There are challenges if one wants to treat cancer with the immune system, particularly using a vaccine. There are prophylactic vaccines for cancer, such as the HPV vaccine, which will prevent the occurrence of cancer. But once cancer has developed, particularly if it's a solid tumor, such as head and neck cancer or triple negative breast cancer, there are significant challenges due to the biology of the tumor and the concomitant treatments that the patient may be undergoing. Those are, of course, with things like chemotherapy and radiation, the patient may have a compromised immune system and may not be able to respond very well to a vaccine. That would be also like the elderly population that doesn't respond very well to the flu vaccine. Secondly, many solid tumors are not presenting known or very active antigens to the immune system. So the immune system is not able to discriminate the tumor from self. So they're not recognized. They're what are called cold tumors. Thirdly, within the tumor, I remember attending a lecture from Dr. Judah Folkman at Harvard years ago, and he made the remark that tumors are like wounds that do not heal. So a wound is a very immunosuppressive environment. Basically, there is a lot of inflammation, and with that inflammation, there are feedback mechanisms that cause immunosuppression. And so within the tumor itself, immune effector cells have a very hard time because the microenvironment is immunosuppressive. And the third challenge, which is where the personalization requirement comes in, is that each tumor or each patient's tumor is unique. So Steve Rosenberg at National Cancer Institute and many others have done decades of studies trying to identify neoantigens that can be used to develop off-the-shelf vaccines. This is now essentially deemed to be not feasible. So to manufacture a vaccine for a patient, it has to be personalized. Currently, we now have, as of about the last 8 years, very effective in some instances, less effective than others, and those are the ones that we want to focus on, a new class of drugs called immune checkpoint inhibitors. The two leading drugs in this category are against a molecule or a protein called PD-1, and they are the pembrolizumab drug from Merck and the nivolumab drug from Bristol Myers. And Indian companies here are in late stages, I understand, of developing biosimilars. So hopefully there will be more access to these drugs in the near future. These drugs act by amplifying a pre-existing T cell response against the tumor. So the caveat to that is if a patient doesn't have a pre-existing immune response, these drugs tend not to work well in those patients. And that's what you see here on this slide. So what you're seeing here is 3-year survival for solid tumors that we are focusing on as sort of lead indications for our product: triple-negative breast cancer, head and neck cancer, non-small cell lung cancer. So even though these anti-PD-1 drugs are approved, what you can see on the right-hand side is the 3-year survival is very marginally higher than with standard of care. And the reason for that is that the tumors are not being recognized by the immune system, so there is no pre-existing T cell response for the drugs to act on. That's where our product comes in, and it incorporates our proprietary technology that we call biological adjuvants. And these differ from common adjuvants that are used in vaccines, such as alum salts, saponin, and whatever is CPGs. Those stimulate broadly an inflammatory response. What we aim to do is to take known immune effector mechanisms, such as those elicited by interleukin-12, B7, GM-CSF, and use those known immune effector mechanisms to drive an immune response in a specific direction, such as, for example, an anti-CD8 T cell response against a solid tumor. And the way that we have engineered this platform, thanks to our Chief Scientific Officer, Dr. Selvaraj, who is from the Chennai region of India and now is faculty at Emory, is to incorporate at the C-terminus of the protein a glycophosphatidylainositol tail. So this glycolipid tail is added to the protein during post-translational processing when these are made in CHO cells, and that then enables the purified recombinant protein to self-assemble in the right orientation into any lipid bilayer. So we are using nanoparticle antigen formulations with antigens to whatever target we are desiring on the surface, along with these recombinant protein adjuvants. This has several advantages. First is when the antigen particle is seen by an immune effector cell, such as a dendritic cell, the cognate receptor on that cell may be activated by the cytokine, such as interleukin-12, to instruct the dendritic cell to go in a particular direction. Secondly, this provides a significant safety margin. So again, coming back to interleukin-12, this drug failed in the 1990s due to toxicity. So with our product, we only have very, very tiny amounts of IL-12 that are required. It acts locally, and so it can be used very safely. And thirdly, this is a modular platform. So it is possible, if someone has an idea about autoimmune or other indications where different cytokines or immunomodulatory proteins would be required, they can be engineered and incorporated onto particles in the same way that we have here to go after cancer. Our cancer product that we call Membrex is comprised of the following 3 components. One component is derived from the patient, that's on the left. And the important thing here is we never need to culture cells or make viral vectors. We acquire at surgery or biopsy a sample of tumor, about 1 gram, and this is frozen at the site and shipped overnight to a central lab where nanoparticles are made by a mechanical physical process. And these nanoparticles are essentially fragments of the plasma membrane from the cells in the tumor. So on the surface of those membranes are all of the neoantigens and tumor-associated antigens from that individual patient's tumor, including carbohydrate antigens and antigens that are not just class 1. So if you're trying to identify neoantigens using sequencing, for example, the only AI approaches right now to identify neoantigens are limited to class 1 neoantigens. So we can actually capture the class 2 as well on the surface. So we have a very broad array of antigens. These particles are then loaded with the biological adjuvant proteins that we've already manufactured in bulk in CHO cells to produce the final drug product that's about 97% tumor membrane protein, 3% cytokine protein. The manufacturing process can be done in 2 days, producing multiple doses of vaccine that can be stored frozen, shipped back and stored in the hospital pharmacy and administered intradermally to the patient on an outpatient procedure. So this is much simpler than a cell therapy approach, much quicker, much lower cost. I've already touched a bit on the effector mechanism. This is just a theoretical slide of the mechanisms we are targeting. So, of course, innate immune system, natural killer cells, both CD8, which is a B7-1 target, and interleukin-12 would be stimulated. And then that would cross-stimulate dendritic cells to drive at the end cytotoxic T lymphocytes or CD8 T cells to attack the tumor. The non-clinical studies that we have performed, I'll focus on head and neck cancer. We are in discussions now with hospitals, medical centers in India. We are in phase 1, have approvals in the US, and are looking to come to India to expand clinical development. Head and neck cancer will be our lead indication. So today I will focus on the non-clinical data in head and neck cancer. So I will show 3 models. The first is the easiest to treat. It's a squamous cell carcinoma model called SCC7. And the important thing here is when the vaccine is given to animals with existing tumors as a single agent, 40% of the animals survive long-term, and 100% of those animals that survive long-term are resistant to re-challenge with tumor. So we are providing durable protection against the tumor in 40% of the animals when the vaccine is given as a single agent. Of course, 60% is a failure, but if you compare this to any other immunotherapies given as a single agent, this is very good. And clinically, almost all of these types of therapies will be given in combination. The next model is a model that is representative of the minority of head and neck cancer patients that respond to anti-PD-1 drugs. This is called the murine oral carcinoma 1 model. So this is a syngeneic mouse model, and these are published data from Dr. Bomaretti, who is a professor at Emory University that works with Dr. Selbreis. So both the next 2 slides that I will show, it's the same setup. We have animals with existing cells, tumors, and we have prepared from tumors of parallel animals, membrane vesicles from the MOC1 tumor, and then loaded onto the surface, in this instance, the murine B7-1 and murine IL-12, because we need to have homologous proteins to be active in the mouse. And they are then administered either alone or in combination with anti-PD-1 monoclonal antibody. On the left, one can see in the green bar, or the green line and the red line, that both the anti-PD-1 monoclonal antibody and the vaccine alone are having an effect on tumor growth. When they are combined, we can see by the blue line that there is a significant reduction in tumor growth. But again, more importantly, looking to the right side, this is what's important if you're a patient. On the right side, all the animals that got monotherapy, whether it was the Membrex vaccine alone or the anti-PD-1 monoclonal antibody, died. 40% of the animals that got the combination had a durable effect. So this is what is important if you were a patient. We hope that this is what we'll see clinically. The last slide on efficacy data is in the murine oral carcinoma 2 model. This is a model that does not respond to anti-PD-1 monoclonal antibody. So this would be representative of the majority, 85% of patients that don't benefit from the current immune checkpoint inhibitor drugs. And so here is what I mean, and I want to show, I think this slide clearly demonstrates why one needs to initiate a T-cell response against the tumor. So on the left-hand side, we have data with single-agent administration of the Membrex vaccine to the animals. And we can see that the green line the tumors are growing more slowly, but they're still growing. So, okay, the tumors grow more slowly, but if I'm a patient, my tumor is progressing, that's not a good outcome. But immunologically, important things are happening. So we administered the vaccine subcutaneously, and that subcutaneous administration of the vaccine caused T cells, CD4 and CD8 T cells, to infiltrate into the primary tumor. So it trained the immune system to find the tumor and attack it. It also set up a systemic immune response against the tumor such that metastasis in the lungs was significantly reduced. So now that we've established a T cell response against the tumor, if we look at what happens when we combine this with anti-PD-1 monoclonal antibody, you can see the black line on the right-hand graph. There is very strong synergy. Essentially, we turn tumor growth off. And that's in contrast to the red line, which is the anti-PD-1 monoclonal. It doesn't work at all if it's given alone. So this really shows why you have to have a pre-existing T cell response for these drugs to work, and that's what we aim to accomplish. We've performed GLP toxicology studies as part of our submission to FDA. We did those studies at doses above the estimated efficacy dose by 5x, with and without checkpoint inhibitor drugs, both ipilimumab and pembrolizumab equivalents. And we also did them in normal animals and in tumor-bearing animals. Most importantly, we did not see any evidence of autoimmunity, and we did not see any evidence of IL-12 toxicity, which is known to be a very toxic cytokine when given systemically. So based on the data that I've shown you, we've been cleared to proceed with phase 1 trials in the FDA and the US, and we're looking to come to India to expand development. Manufacturing, I've already touched on this a little bit. I'll just show how it works clinically. Again, the surgeon would acquire a small sample of tumor that would be shipped to a central lab. The membrane vesicles are manufactured. That takes 1 day. The next day, the pre-manufactured biological adjuvants are added to produce the final vaccine product. Currently, QC takes 16 days because we do a USP71 sterility test on every vaccine, and that's a 14-day test. So in theory, once we get all of this in late stage, we believe that we can do this in less than 1 week. But 3 weeks, which is our current manufacturing and release time, this compares to 6 to 8 weeks for a personalized mRNA vaccine and about 4 to 6 months for many cell therapies. So we're much faster, and this is important when patients have rapidly progressing cancer. Talking a little bit about how we QC the membrane vesicles. So this is a representative batch of breast cancer samples that we processed, and these are known breast cancer antigens on the surface of the membrane vesicles. Importantly, we processed 16 triple-negative breast cancer samples and 4 HER2 positive breast cancer samples. And so what you can see, I apologize, what you can see in the purple scatter plot, this is HER2, and the HER2 positive samples are represented by squares. So we're at least semi-quantitatively recovering the known tumor antigens on the surface of the nanoparticles. And then the triple negative samples, which should be HER2 negative, they were scored that way by the pathologist, they're all showing very, very low levels of HER2. And then the relatively new breast cancer antigen TROP2 is present in varying amounts on all the samples. So this is one of the tests we do. It's a flow cytometry test for each TMV sample. Now looking at head and neck cancer, this is essentially flow cytometry data showing that we have loaded successfully onto the surface both B7-1 and IL-12. So we have unstained TMVs. TMVs have been unincorporated. You can see there is some varying amount of IL-12 in the tumor, but on the bottom, these are 4 different samples from patients at Emory. You can see after loading the IL-12 and B7, we have very consistent loading of the cytokines onto the surface of the TMVs. And I will mention that the glycolipid mechanism for attaching the proteins to the surface is present for 400 some odd membrane proteins in the human body, It's also a mechanism that's present across eukaryotes, including in plants. So this is a natural mechanism for non-covalently but stably associating proteins with the plasma membrane of cells. I have just 3 more slides. The first is what we're planning for clinical development. So our first focus for head and neck cancer will be patients that have recurrent tumors. So this is a very, very significant issue for these patients because typically they have had neoadjuvant chemoradiation therapy and then surgery. And because of the anatomical location of the tumor, they may have already had some disfigurement associated with the surgery. So when the tumor recurs, it is a devastating diagnosis because it means that it's usually resistant to chemoradiation therapy, and it's usually in an anatomical location where getting clean surgery to excise all of the tumor is almost not possible. So this is a very high need patient population and one where checkpoint inhibitors are being used, but with not very much success. So this is where we choose to focus. Phase 1A will be to establish safety and immune response, and Phase 1B or Part 2 will be in conjunction with checkpoint inhibitor, and then we'll look at the data and go from there. I want to talk about one other aspect of the technology, and that is it can be used for other applications in immunotherapy. So we were visiting with some researchers in Genome Valley yesterday, and they said we have a lot of T-cell companies here working on tumor infiltrating lymphocytes. So one of the challenges, in theory and has worked well, but one of the challenges for preparing an autologous TIL therapy, that is to take a tumor sample, isolate the infiltrating T cells, and then expand them and give them back to the patient, is when those T cells are being expanded, they're being expanded in cell culture in a bioreactor, and they lose their orientation. So they forget where they need to go when they are reinfused into the patient. And so in collaboration with Dr. Ned Waller at Emory, who's developed a specific drug cocktail, some experiments were performed and just published last year in a patient-derived tumor model of colon cancer. And I want to point you just to the lower yellow or orange diamonds on each graph. These are the tumor growth rates in the animals that received T cells that were cultured in the presence of our Membrex vaccine with the drug cocktail. So there's an additive effect. So this can be also used in the manufacturing of other immunotherapies to train them to attack the patient's tumor. So we are very interested to develop collaborations in this area as well. Our portfolio in oncology are booster vaccines for long-term maintenance of the immune response. And then we have other applications, including the T-cell application that we are working on preclinically. And we'd be happy to talk to anyone who may be interested about working with us on some of these other technology applications in addition to our lead drug. I want to thank certainly Mr. Shaker Reddy, who's a company founder and is here with me, and is a native of Hyderabad. So he is showing me all of the finer parts of Hyderabad, including the cuisine, which I am very fond of. Dr. Christopher Pack, who's head of our preclinical group, Sam Ramachandran, who has led all of the manufacturing efforts, and Dr. Kristen Jacobson, who handles a lot of our regulatory affairs and analytical. On the Emory University side, certainly recognizing our Chief Scientific Officer and founder, Dr. Selvaraj, his collaborator, Dr. Bama Reddy, the lead clinical investigator in head and neck cancer, Dr. Dongmung Shin, and our collaborator on the T-cell experiments, Dr. Ned Waller. So with that, I'll take any questions that you may have, and thank you for your attention.
Good afternoon, sir. It was a very informative presentation regarding the vaccine. As you said, actually, the Membrex vaccine. Is there any mutated cell specificity for this one or not?
No, sir. These all the the membrane nanoparticles were prepared from growing tumors that were harvested, frozen, and then they were processed by the same process, whether it was for animals or for patients. So the mutations that would be present in the tumor would be the naturally occurring mutations for that tumor. if I've understood the correct question correctly.
What about its success rate? It is used as a monotherapy.
As a monotherapy, I did show in the first animal model that we had 40% durable control as a monotherapy. I believe clinically the single agent will be most active in patients that have minimal burden of disease. So one may envision, for example, a patient with bladder cancer, which has higher frequency of recurrence, or triple negative breast cancer. They have had resection with clean margin, but they don't have a pathologic complete response. And they have low T-cell infiltration into the tumor upon pathology assessment. In my mind, those patients would be the perfect patients to vaccinate in the adjuvant setting when they have low burden of disease. So I think there is possibility for single agent activity, particularly in those patients.
Yeah, thank you very much, sir. It was a nice work and presentation. Whatever the vaccines you are giving after developing from the cells obtained from the cancerous cells, they are working at all the levels of cancer stages of cancer of particular organ, and they work throughout the other organs of the body also.
So we have modeled this we of course don't have clinical data yet to inform what I say. Based on the animal models, we have administered the vaccines in animals that had small tumors, and then I didn't show for time triple negative breast cancer, but these data are published in the 4T1 breast cancer model at such a time that the animals already have lung metastasis. And what we can show is that we have additive effects with checkpoint inhibitor drugs in animals that have established lung metastasis. So essentially stage 3, stage 4 cancer in the animal, we have activity.
So theoretically it will work, theoretically. Practically we have to establish it, but according to you, theoretically it will work throughout the body.
That is correct. That is correct.
Thank you.